These are going to be your cellular respiration notes part 2. We're going to start with aerobic respiration. Aerobic respiration means respiration with air, and it has 3 major steps but many smaller reactions. So we're going to walk through the 3 main steps, and I'll kind of give you a little bit of detail on the smaller reactions as we go. The first step is glycolysis. As you can see from both of these images, glycolysis occurs in the cytoplasm, and it's where glucose is going to be broken down. The second main step is the Krebs cycle, and this is going to occur in the mitochondrial matrix, and it requires oxygen to take place at this point, whereas glycolysis can be anaerobic, does not require oxygen. The Krebs cycle and the electron transport chain both do require oxygen. And then the third step is phosphorylation, and that occurs in the electron transport chain, as it's written here as the third step. So I'm going to walk you through these 3 steps. I think these images are very important to keep in mind while we talk about it. So in glycolysis, again, it's the breakdown of glucose in the cytoplasm. It is anaerobic, which means it does not require oxygen. You have this big fancy formula, C6H12O6, which is simply glucose. And then these are the molecules coming into it that are going to make the energy molecules. One of the things that glycolysis makes is a little bit of energy in 2 different ways. It makes ATP and it makes NADH, which is an energy carrier. So you have to, in order to make ATP, you have to bring in 2 ADP and 2 phosphate, and then 2 NAD+, and that's going to make, total, it's going to make 2 ATP and 2 C3H4O3, which is basically pyruvate. It's the new molecule. So we've taken a 6-carbon glucose and broken it into 2 3-carbon pyruvates. And then we also make 2 waters in this step, and then we make 2 NADHs, which will go in and help do the next step in the Krebs cycle, and 2 hydrogen ions. Glucose is a 6-carbon molecule, as I said, and it breaks into 2 pyruvates, which are 2 3-carbon molecules, and those will enter into the mitochondrial matrix. If you look at this first diagram on the right, I have it, it hardly ever zooms in when I want it to. We have glycolysis, and this arrow, it changes the acetyl-CoA, so pyruvate will come into the mitochondrial matrix, and it'll get changed right before the Krebs cycle into acetyl-CoA, and then it can be used in the Krebs cycle. But as you can see, glycolysis also makes some ATP. And if you look at this arrow coming up and across these electron carriers to the electron transport chain, that's that NADH that the glycolysis is also making. So that's where everything goes. Water will leave, ATP gets stored in the cell, pyruvate gets moved to the mitochondrial matrix and makes acetyl-CoA, and we've made some electron carriers for our electron transport chain. And the main thing we did when we broke down the glucose is we changed a 3-carbon glucose into 2 pyruvates. This gives you quick energy. It's like your turbo button. If you can remember in class when we did the ball squeezing activity, and it burned, it's because we were having to make lactic acid to make that quick energy, make that turbo button through doing a lot of anaerobic respiration, and we'll talk about why that is and why that burns in a later video. But you're making a lot, you can do this quickly, to help make a lot of energy very fast. But it does require 7 steps in glycolysis itself. So while I say there are 3 major steps, each major step has many smaller reactions in order to take place. Our next step that we're going to talk about is the Krebs cycle. So after we make our pyruvates, we go into the Krebs cycle. The Krebs cycle has a lot of steps as well, but in the mitochondrial matrix, before the Krebs cycle, pyruvate gets broken into acetyl-CoA. It gets broken down by a pyruvate dehydrogenase complex. You don't need to know that, but it does get broken down into acetyl-CoA. And in the Krebs cycle, this is basically where enzymes will break down organic molecules, and they will break them into carbon dioxide. And carbon dioxide is a waste product that leaves the cell. So how does that happen? It says pre-cycle, pyruvate is broken into acetyl-CoA, which is a 2-carbon molecule. So you're losing a carbon right there. So carbon dioxide, if you look up here, it says acetyl-CoA, and it's got carbon dioxide arrow, or an arrow leading down to the 6 CO2s. One of that comes every time we lose a carbon, it changes into carbon dioxide. So there's one of those carbons. And then acetyl-CoA will go through a series of different reactions, and it will lose another carbon and then another carbon, to where eventually from the 6-carbon glucose we end up with 6 carbon dioxides that have to leave the cell. So the goal is to release carbons in the form of carbon dioxide, CO2, and we want to keep from the glucose, we are keeping the H+ ions and the electrons. Remember, a hydrogen is just a proton, which is represented by H+, and an electron. So we want to keep those while we get rid of the carbons, because we no longer need them. And we want to use those in the electron transport chain, and we're going to do phosphorylation in that step. So what happens to those hydrogen ions and the electrons that we want to keep and make is we're making more electron carriers. So H+ gets added to NADH and FADH, or FADH, yep. And so if you look in your Krebs cycle, it's got a couple of arrows that are going up to that second line at the top, and those go into this electron carrier box. So we are making NADH and FADH electron carriers, and we're taking those off of that glucose, and you can also see it in this next diagram. There's the NADH and FADH being made, and we're also making just a little bit of ATP here, about 2 ATP, but we are making electron carriers to be used in that next step of cellular respiration, aerobic cellular respiration, I should say. So our final step is called the electron transport chain, and this is where phosphorylation occurs. So step C is phosphorylation. It's where you add a phosphate to ADP, and you can get as many as 36 ATP in the mitochondria. 36 ATP in the mitochondria. High energy electrons are donated from NADH and FADH2 into the electron transport chain. And you can see this in this pretty messy diagram, but basically high energy electrons are coming off of NAD and FADH2, and they are being pushed through these proteins in the cell membrane. They're being pumped through, pumped through, pumped through. The electrons are this yellow arrow that's coming through these proteins, and that's what's pulling the positive protons through. And then it says H+ ions are pumped through ATP synthase to generate a lot of ATP. So after this ion gradient is formed, all these positive ions that got donated from NADH and FADH2, they were pulled through by that electron. They're all built up over here, and we have an ion gradient, meaning there's more positives on one side than the other. Well, the only way that they can go through this phospholipid bilayer that repels charges is for them to travel through this protein called ATP synthase. And as it comes through, phosphorylation occurs. So a phosphate group is going to be added to ADP, and it's going to create ATP. And that little lightning bolt there represents that energy that's being stored in that final bond. So let's see where I left off on the notes. Okay, it says oxygen enters lastly as O2. So oxygen comes in the form of oxygen gas, or O2. And it will accept the hydrogens and electrons to form water, which will leave the cell as a waste product. So where did those hydrogens go that come through the ATP synthase, and where did those electrons go? Well, they're attracted to the oxygen, the O2. So oxygen will kind of split in half. Each O2 will make 2 waters, and you will get 2 hydrogens from this process, 2 electrons, and you make waters. So each O2 makes 2 waters and pulls off 4 hydrogens total and 4 electrons total in order to make that happen. So basically hydrogen gets put back together after it was pulled apart back when we took it off of the glucose molecule, and it gets created into water in this last step. And that's very important. Without oxygen coming in, the electron transport chain, the ETC, and the Krebs cycle will shut down. So then there will be no more big energy made, which is why you would go into fermentation, which will be in the next video.